I'll be more interested in the fermentation part. That is the chemistry involved in the process.
Yeah, well, fermentation and also wine aroma.
Yeah.
Don't tell her.
Yes.
Don't tell her
Yes.
about the aroma.
And we have students here who are doing chemistry as well. Most of the students, they are doing botany, zoology, and chemistry. So,
Okay.
you know, the topic, so the topic is like multidisciplinary and it is for everyone's usage.
Yeah. Has he come? No, he's coming. Sorry, Angelika, we stuck with some joining in problems. Yes.
Okay,
Yeah.
it's not a problem.
He's head of
It's
our
so wonderful that technique makes it possible to
yes,
do
yeah, we
such
could
a
do this,
seminar,
yes.
but it still has
Absolutely.
problems.
Yes, very true.
And you know, like before this lecture, I didn't know that do we have these courses in our country, and I realized almost every agricultural university offers this as a full paper. So meaning it was we are adventuring into something new, and which is very good for our students especially.
I think it's the positive outcome of the pandemic.
Yeah. So we are waiting for him. Do I call him again?
And I think it's always nice to for the students to see the spirit behind the work.
Yes. Yeah, sir is coming. Good morning, sir.
Good morning, sir.
Yeah.
Good morning, sir.
Yeah.
Good morning, sir. Good morning, sir.
Good morning.
This is Dr. Angelika, and this is Professor Vipin Agarwal, our principal, head of the institution.
Nice meeting you.
Nice to see you. Thanks for the coming. Thanks.
It was a pleasure.
My pleasure too.
Yeah.
So Manju, can we start it?
Yeah.
Yes.
Yeah. So we start this, and so yeah, I think we are 4 minutes late. My apologies for that. Yeah. Good morning, good afternoon, and very good day to everyone. Very warm welcome to this webinar on the science of viticulture and enology by Dr. Angellikaa Scartl from Bavarian State Institute of Viticulture and Horticulture, Germany. Before we formally begin with the talk, I would request our principal, Professor Vipin Agarwal, to welcome our speaker for the day and share his words of wisdom with the audience. Sir.
So good afternoon, friends. It's my great pleasure and it's a privilege for me to welcome Dr. Angelika for this webinar. I'm sure his words will greatly inspire our students and will know many things which we may not be doing in India. So I thank you on behalf of the Delhi University, on behalf of Sri Aurobindo College and Department of Botany of Sri Aurobindo College. I thank Dr. Angelika for sparing her time and sharing her thoughts with us. And I'm equally thankful to Department of Botany for giving me this opportunity to interact with Dr. Angelika this afternoon in India. These type of programs, when we organize, these gives motivation to our students that how things are happening in the world. It motivates the students to learn new things. And apart from the teachers, if people from different part of the world speak something that gives that motivates students to understand the topic in a more systematic manner, because some of the things which we may not be doing in India, in that part of the world, they are doing it and they will come to know all these things. And this is always a good thing which we take from adversities. Because of this COVID-19 throughout the world, I mean, we say altogether a new world in the sense that up till now or before this COVID-19, we used to meet physically and used to interact with each other. After this COVID-19, and in that scenario, calling international expert on a topic for a lecture in a college was a difficult thing. Whenever we used to organize seminars at that point of time, normally international experts used to come, but for a particular talk of an hour or so, international experts finds difficult to come to India for a lecture of 1 hour or like that. This corona has taught us that we can do this online also, we can do it in the virtual mode also, and we are doing a lot of these type of webinars in India and in our college. So once again, I would like to congratulate Department of Botany for organizing this webinar, Dr. Manju Gupta, the teacher in charge of the head of the Department of Botany, Dr. Sangeeta Kaul, our IQAC community, and all the organizing committee members. And my special thanks are to Dr. Angelika for sparing her time and sharing her thoughts with us. With these words, I hand over Dr. Manju to Manju. Thank you very much.
Thank you.
Thank you, Vipin sir, for your encouraging words. Before I ask Angela to start with her talk, I think it makes sense that I formally read her short profile. Dr. Angellikaa Scartl did her education in biology and chemistry from Justus Liebig University, Germany. She did her PhD on hereditary cancer, working on a specific genus of fishes, Zyophorus. How do you pronounce that? Zyophorus. Yeah. She was a visiting scientist at Hopkins Marine Station, California. Her present interests and specializations are grape breeding, transgenic grapes, detection of grape pests, truffle cultures, and molecular detection of mycorrhizal fungi. Today, she'll be talking to us about viticulture and enology. I understand the topic is very interesting and there is a lot of enthusiasm among students who want to pursue a career in wine making. I find this worthwhile to mention here that this program is specifically organized by the students and conducted by the students. I would encourage everyone to write their queries in the chat box. In addition, there is a brief session of live conversation of the students with Dr. Angelika after her presentation is over. Please keep the fire on. And without any further delays, I would like to invite Dr. Angelika to start with her talk. Dr. Angelika.
Thank you, Manju. It's a pleasure to present you. Let me see. Oh, I cannot share my screen, Manju.
Why? You let me see. You must be able to share.
It says that the host doesn't allow me to
just
share.
Give me 1 second. Sorry, that's all panelists. I think now you'll be able to do it.
Okay. Yeah, here we are.
Yeah.
And there we are with the presentation.
Yeah.
Okay, get rid of this.
Sure.
Okay, I will start my presentation at the view I almost have out of my window, and it takes you to the city of Würzburg, and you can see there is some viticulture about below this wonderful castle here in Würzburg. I want to introduce you to viticulture and enology. And okay, let me see. There is my next slide. Würzburg is right in the heart of Europe, and the wine growing region where we are is called Franconia. And we have a specialty in the wine, which is this bottle. It has a patent and it's only used for a special cognac and for the Franconian wine. All the vineyards are along the Main River. And before I start, I want to give you some numbers. Here you can see the vine growing regions worldwide. And when vines are grown, it's not only for wine production, but it's also for the production of table grapes and dried grapes like raisins and sultanas. So 7.4 million hectares have been under vines in 2018, and it's a production of 77.8 million tons of grapes. So wine production numbers for the world and for Germany. Worldwide, 292 million hectoliters of wine were produced in 2018. In Germany, 0.3% of German crowns are vineyards. It's an annual production of 9 million hectoliters of wine, which are approximately 1.2 billion bottles of wine. Germany is the 8th largest wine producing country in the world, and it accounts for almost 2/3 of the total worldwide wine production. Okay, here is the outline of my presentation. I will talk about the work in the vineyard and about making wine. I will talk a little bit about Vitis vinifera, the grapevine plant and its physiology, about grapevine pests, about the work from harvesting to fermentation, and about tasting wine. And I thought it would be a nice summary to take you through the year of a wine grower. So let's start with Vitis species are distributed in the Central Americas, in Europe, Middle East, and in Northern China and Japan. They're native Vitis species. Some of them are used for making wine, but the wine grape is the European grape, or the wine-bearing grape, as the Latin name says. The plant in the wild, the plants grow on trees, and here you can see a grapevine in the crown of a pine tree, and this leads to special characteristics of the plant, which are apical growth, prominent growth on the tops, and they need a support to bring their leaves to the sun to make photosynthesis. Here you can see the year of the grapevine, and this slide is for moderately climate, and as you will see later, we also need some or quite some precipitation for wine growing. And this is for our region. So we have the growth is here. So you can see the vines start growing at the end of April, May. You can see the bud break, and then the shoots grow. They flower at the end of May, and then the barrows develop, and in September, October, there will be harvest, and in November, the leaves fall down and the buds get ready for bud break in the next year. We have a dormancy of the buds. The buds for the next year are already produced in this growing season. They have dormancy, pre-dormancy, where they are still developing, and then a correlative pre-dormancy where they are not yet ready to break open. And we are talking now about the next year. So what's done in the vineyard this year will be good for the wine in the next year. This is very important for the wine growers. Okay, we have a special type of wine which is only possible to produce in the northern or more cooler climates, which is called ice wine. I want to tell you about this specialty because the grapes have to be harvested when the temperatures are still below 7 degrees, -7 degrees centigrade. So this wine is very, very sweet, and it's very, very special, and it's become more and more special with climate change and global warming. Here you can see the pictures, how it looks in the vineyard. You can see the vine climbing along the wire and the buds are ready to break open. Here there are the first leaves that you can detect. Leaves flower in clusters and they have a specialty because the petals don't open like the petals of a normal flower, but they fall off the flowers and they you have an open flower with removed petals. You can see here the stamina, and here you can already see fertilized flowers. Grapes are 80 to 90% self-fertilizing, and fertilization takes place before the petals fall off the flower buds. Here fruiting has started. You can see the little green berries. They will develop over the year and then will turn into ripe clusters at the end of summer, beginning autumn. There are numerous, many grapevine cultivars cultivated all over the world, and they have different characteristics and different specialities. Some, like Chardonnay, are grown worldwide, others are locally grown. Silvaner is a cultivar which is very prominent in my wine growing region. And just to show you some different characteristics, both grapevines you can see here are Silvaner cultivars, but one is a premium clone with a very low yield, clusters which don't have much berries, and on the other side you can see a clone which has a very high yield. As I already told you, vines need support, and the most common support system is the trellis system that is used in our vineyards. And you can see here is the last year's shoot, and the buds that were produced last year resulted in these shoots, and they will carry the cluster. And how this looks live, you can see here on these pictures. And you can see something which is also typical for our region. There is a green cover in the vineyards, but the green cover competes with the water that is available. So in our region, we cannot have the whole vineyard covered by a green cover. We need to break open every second row to have enough water. And this is from, it's not allowed to water the vines in the Franconian vineyards till now, but they are doing experiments. So you can see here irrigation for this irrigation experiment. The wine growers might be allowed to take water out of the Main River. Okay, let's see. Growth requirements, you've seen it's from the Mediterranean. So the grape wines are adjusted to a warm climate. They start growth at about 6 to 8 centigrade. Optimal growth temperatures are between 22 and 28 centigrades, and they stop growing when it gets too hot. Above 45 centigrade, there will be no growth because the stomata will be closed and no photosynthesis will take place. We have cold winters here, so we can have frost damage in the winter. So the dormant buds and the wood, they can bear temperatures between -20 and -30 centigrade. And when the buds already are ready to break, we can have late frost in spring, and it should not get below -3 centigrade. We had a severe frost damage in 2011. The 5th of May, there was a very cold night where temperatures dropped to -5 and -6. And many vineyards were damaged. Sap flow starts at a root temperature of 7 centigrade. To ripen, the Crepe Berries need 1300 to 1500 hours of sunshine, and they need approximately 690 millimeter annual precipitation. It's in Würzburg last year, we had, or the last 2 years, it was very dry. We didn't reach the 600 measure, but this year we already had a lot of rainfall. So to summarize this, grapevines thrive best in climates with warm summers and cool, rainy winters. Here I have some pictures of frost damage. You can see here the brown parts, they have died because of frost. And here you can see on a microscopic image how the ice crystals have destroyed the walls of the leaf cells. And you can also see frost damage in the buds, which is important for the winters. So they control the buds for frost damage. And you can see here in the bud, the center of the bud, where the meristema are, is destroyed. But because the grapevines often have damaged buds, they have a security system. So besides the main bud, there are 2 additional buds which start growing when the main bud is destroyed and starts and stops spreading hormones. So this bud is still green and it will grow and maybe make sure that there will be a harvest. And here you can see a cut to a healthy or not frozen bud. It's a cross section, and here you can see the green bud. Okay, I've talked about frost, and there are other abiotics and biotic pests that stress the vines. I talked about water, so water stress, and also with global warming, sunburn has become more and more prominent. Some vintners started to even put white foils into the vineyard or to cover the grapes when after a bad weather period, the sun comes out again. And here you can see grapes that have both not enough water and sunburn. And you can imagine that it will not be good for winemaking if you will harvest these clusters. There are fungi that attack grapevines, and this is a very severe problem. We have 2 types of mildew, the downy mildew and the powdery mildew. You can see here the leaf symptoms. And the fungi also grow on the berries. Here you see the downy mildew symptoms, and here is a cluster with powdery mildew. These cannot used for wine production. And there is also another fungi, it's the noble rot. In some areas, it is used, these berries are used for winemaking and create special taste, but in our vineyards, we don't want it. The taste is not wanted in Franconian wines. So to prevent fungal disease, there has to be plant protection. Here you can see the vineyard is sprayed. In our region, the powdery mildew is the main problem, but with global warming, downy mildew is more often also. In Franconia, the grapevines are sprayed approximately 8 times a year. In other regions, up to 16 applications of fungicides, which is quite a lot. and application has to stop when the berries start to ripen. So the plants have to be protected when the ripening process or the veraison, when the berries start to soften and they get their the colors they will get when they are ripe. Besides spraying against fungi, there has been very successful breeding for fungal resistance. It started in the 1960s. There are a lot of wild species that are resistant against these mildew fungi, and there are already new varieties with good qualities. They are mainly used in organic wine growing. There are insect pests. You have heard of the vine louse or of phylloxera. Here you can see some pictures. In the center, it's the winged form, and they do galls on the leaves. This is the under the leaf, you can see the galls, and if you open them, you can see here this form, which is laying eggs into the galls, and they can also be on the roots. You can see here is the phylloxera and the eggs, and here you can see how the roots are damaged. So it's very, very bad. It's fatal to the vines. The vines die, and we have a special form of biological pest control. All the vines that are grown in European vineyards have to be grafted on phylloxera resistant rootstocks. Here the grafting is still done by hand. You can here see the rootstocks and the shoots of the Vitis vinifera, which are connected by this machine. And when the wine grower buys the wines, they look like this. The root is formed by the phylloxera resistant plant. And here you have the buds of the Vitis vinifera cultivars that are put on top of the rootstocks. Now we come to the Crepe Berries. Here you can see a cross section with the seeds. Some or most of the wine grapes have seeds. It's only for table grapes important to have seedless grapes. You can see the different sections, the endocarp and the mesocarp, and the endocarp and the mesocarp have a lot of juice and a lot of sugar, and the exocarp, the berry skin, which can also have different characteristics. And you can see the brown dots. These are lenticels, which were stomata and now have cork substances in here in the cells. And here you can see the ripe berries. You can see the wax on top of the berries, and these are about to be ready to harvest. So ripening of the grape berries, you can check chemical properties of the berries. Of course, we also can do it by tasting and by feeling. The berries get soft, of course they get bigger. And what's happening inside the berries is that acids are produced and sugar is put into the berries. So when the berries grow, you have a high acid content, as you can see in the blue curve. It's mainly two acids. It's malic acid and tartaric acid. So it's the apple acid and the wine acid. And with ripening, the acids go down, and the sugar in the third phase, the sugar goes up. To determine the best point for harvesting, the wine growers measure the sugar content. In Europe, we measure the Oechsle, and they start harvesting for table wines at about 80 degrees of Oechsle, which means 160 grams sugar per liter. But for high quality wines and sweet wines, the sugar content can go up to maybe 120 Oechsle. And I put the number here, 80 degrees Oechsle make 10.6% alcohol in the fermented wine. So here, this is an optical measure. The berry is pressed. You have it's a refractometer, you know, maybe from the lab. The grape juice is on here, and you look through here, and it's measured how the light is broken by the sugary liquid. Harvesting. So when the grapes are ripe, they have to be harvested. And here you can see a typical steep Franconian vineyard. And in the back, as Manju mentioned, the truffles. This here is the truffle plantation in the background. This is mechanical harvesting, which is more complicated, of course, in the steep hills, but it can be done. It's done for table wines and lower quality wines, but high quality wines are still harvested by hand. Now we come to the process of winemaking. After the harvest, the grapes are pressed. The must is put into tanks for fermentation, and then the wine stabilizes and it's filled into barrels for resting, and then later the bottling occurs. I will tell you about the time frame when we do the summary of the wine growers here. For red wine fermentation, this is typical for white wine fermentation. Red wine fermentation is done a little bit different. So here you can see harvesting by hand. The grapes or the clusters are put into this machine. They are destemmed because the fermentation will take place in the beginning of the fermentation will take place in the mash. So here you can see the berries without the stems. If you leave the stems in the fermentation, you will have a lot of tannins in the wine, and this will not taste good. The fermentation in the mash will maybe last for 1 to 3 days, and then the mash is pressed and the must is put into barrels for the rest of to finish fermentation. In our wine growing region, it's not common to stop fermentation. So fermentation stops when it's not much residual sugar left, and if the alcohol content is so high that the yeast cells die. So we have a alcoholic fermentation, which is done by the wine yeast, Saccharomyces cerevisiae. It's also the bread baking yeast and also the beer making yeast. It's the same species. There are a lot of different yeasts. If you check lists, you can buy purebred dried yeast, which is added to the wine, but there are also many yeasts or also different types of yeast on the berry skin. So if no purebred dried yeast is added, the wine or the must will undergo a spontaneous fermentation. And this is something the wine grower can not influence. So he cannot influence the types of yeast that are on the berry skin, and he cannot put, or very much, he can influence, of course, the temperature of the fermentation, but not what's going on in the tank. So during the alcoholic fermentation, the sugar is metabolized by the yeast into carbon dioxide and ethanol, and of course energy for yeast propagation or the cell. You can see here the cells are budding. And there is another fermentation that is going on and wanted in red wines. It's the malolactic fermentation. And this fermentation is done by bacteria. The green arrows show you the chains of bacteria that do the malolactic fermentation. The bacterium is called Enococcus oenii, and it turns malic acid into lactic acid, which makes a smooth structure in the wine, and also not an aggressive taste of the wine. So red wines have to undergo this second fermentation by bacteria. When the wine is fermented, the yeast cells die and sink to the bottom, and the wine can rest on the yeast before it is bottled into the glass. And here I have I wanted to show you the label. It needs to have, all the red things have to be on the label, and the black things can be on the label. So you have the year of the harvest, of the vintage. Here we have the variety, and wines also can have different varieties. They can be blended. So it would say blend or cuvée, which is the French word for blending. Then you can have attribute for the residual sugar. And here it says dry, so not much residual sugar left. And then we have which type of wine, which quality level of wine. The wine will have a number, because all the wines are tested except the table wines, which are the cheap wines, and a lot of wine growers even don't produce this sort of wine. Then the name of the producer, here it says Walter and Normal Wintner, and here you have the vineyard. The vineyards have sometimes very special names, and it's a PR thing also to have a nice name for your wine. We have the wine growing region. In this case, it's Pfalz, the Palatinate. It would be Franconia for our region. You have the contents of the bottle, and you have the alcohol content. So a lot of information you can read, and some wine growers also put more information on the back, like acid and sugar content. During the fermentation, wine aromatic compounds develop. You have a primary aroma that comes from the grape variety. Really good wine tasters or sommeliers, they can taste the variety of the grape, but it's very, very hard. And you can use special yeasts which make very prominent aromas, and the variety will be, the taste of the variety will be covered. You have about up to 40 aroma substances in the must. And during fermentation, this goes up almost by tenfold. And many, many aroma substances are still not yet identified. You can only see the peaks in the analysis, in the gas chromatographic analysis. The secondary aroma are built during the wine making process, and they are influenced by the method of pressing and also fermentation control. So if the fermentation if the must is cooled during fermentation process, you get very fine aromas, which are clear. I already talked about the yeast. The yeast selection influences the aroma very, very much. Also the material of the tank, you've seen the steel tanks and the barrel. There is a special if you put red wines into oak barrels, the wood taste, the tannins go over into the wine, and you will have like vanilla aroma in the wine. This is typical for red wines with in the barrique cask, it's called. You can have this aroma also by adding oak chips into the fermenting wines, but it's not allowed in Germany. And then you also have tertiary aromas that develop as the wine ages. So we have the influence of the grape variety. We have the the fermentation control here, and we have here the barrels. This is the wine cellar underneath the residence in Würzburg. To describe this aroma, it's very, very hard, but if you deal with wines and if you want to sell wines, the University of Davis in California developed this aroma wheel for the wines, and you can see it's many, many aromas, and some of them are not nice in the wine, like the chemicals aromas over here. The floral aromas and the spicy aromas mainly derive from the berries. And to make this, to show you this in a picture, it will be in German, but you can see all the pictures. So if you taste wine, you have to use your nose, your eye, and your mouth. And if you describe a wine, the eye starts with the color, and with if you have carbon dioxide pearls here, if it's clear or if it's not clear. And then you smell the wine, and it's very, very many adjectives you have here, and all the students have to learn what these adjectives mean while tasting the wine. Some of them, it's very hard to, for someone who isn't trained, it's very hard to say what these aromas would taste like, or if it's fruity or more flowery or woody. So you really have to be trained on a set of single aromas to use all these words. And then we come to the aromas you smell and you taste when the wine is in the mouth. And here you also can see there are aromas you would like to have in a wine, for example, citric fruit or pineapple here, but you wouldn't want to have grass flavor or even fungal or hay, this is here, or tea aroma. And of course, if you look here, the animal aromas, some of them are nice in the wine, but some of them you really don't want to have them, like here, rotten egg or nail polish. It's the esters from the nail polish that are produced by some of the yeasts and wouldn't taste well in the wine. I thought this would give you an overview, or the pictures would give you an overview what someone can taste in the wine. So here are my slides of the wine growers here. And as you can see, he has work to do in the vineyard, in the cellar, and we begin the year in January. The wine grower goes into the vineyards and prunes the wine. In the cellar, there is not much work to do. The malolactic fermentation of the red wines is completed and the maturation process starts, and the very light wines are already ready for bottling. March, April, the vine enters the new growing season. You can see the sap rising, and it weeps out through the cutting scars from pruning. And in the cellar, the wines are bottled, while some wines still continue to rest. I told you that we have a danger of late frost in spring. We have days in May that are called the ice saints. It's typical for our region, for the Northern European regions, to have this late frost. And it shouldn't be too low to damage the vines. In June, the flowers turn into berries and the training begins. The canes are lifted and tied along the wire trellis, and the shoots are topped. In the cellar, the final bottling occurs. July and August, there are measures in the vineyard to improve the quality of the wine. The leaves are stripped to have direct sunlight on the clusters, and surplus clusters are removed to have larger berries on the clusters, and lignification occurs in the shoots. The shoots turn from green to brown. In the cellar, it's preparation work for the new grape harvest, which will start then in September. September, October, it should say here. The grapes are harvested, and it's very busy in the cellar. The grapes are received, destamped, and pressed. Alcoholic fermentation will be started by pure culture yeast, or the wine grower will wait for spontaneous fermentation, and the vinification takes place. In the vineyards, the leaf drops from the vines in November. The vineyards are fertilized with minerals and organic supplements and are prepared for pruning. And in the cellar, the fermentation process is almost finished. Okay, back to the fortress of Würzburg and to a summary of my talk. You learned that Vitis vinifera is a climbing plant with all the characteristics of a climbing plant. It likes warm summers and cool rainy winters. Grapevines are grafted on phylloxera-resistant rootstocks. Best management practices in vineyards include pruning and training, as well as plant protection against drought, sunburn, and fungal diseases. Ripe grapes are harvested, crushed, and pressed for fermentation. Wine yeasts metabolize the sugar of the mast into alcohol. Fermentation and aging lead to the full aroma of the bottled wine. So I want to say thank you with a picture of the Franconian wine queen and also some fancy box bottles. So thanks a lot for your intention, and I wait for your questions.
Yeah. You could stop sharing now, Angela. Yeah.
Yes.
Thank you, Angela, for your such an illuminative talk.
Thank you.
Yeah, I'm sure everybody around here would have benefited from this and wants to make a career in winemaking. For everybody's information, I would like to mention that the subject of viticulture and enology in India is taught in all agricultural universities in every state at undergraduate level, and at Dr. Baba Saheb Ambedkar Marathwada University, Aurangabad, they offer a postgraduate diploma in wine technology. So students take a lot of interest. By chance, I was just, because I got curious about this, and we already have lots of institutes offering these subjects, and people are taking huge interest in these things. So I appreciate and I understand the students would have really benefited.
Manju, you need to unmute yourself. Manju, please unmute.
So sorry for this to happen. I will do it again. No problem. Yeah. So I wanted to say you thank you, Angela. And
you're welcome.
For your such an informative talk. And I'm sure that everybody around here would have benefited from this and wants to make a career in winemaking. For everybody's information, I would like to mention that the subject of viticulture and enology in India is taught in all agricultural universities in every state at undergraduate level. And Dr. Baba Saheb Ambedkar Marathwada University, Aurangabad, they offer a postgraduate diploma in wine technology, because I thought this was a very much interest generating talk for the students here. So for those who want to pursue here, we have this subject available in almost all the universities around. So because this was mostly the student oriented program and they want to have a live chat with you, meaning face to face. So what they have planned is, we'll move further and I'll give this to the students they want to join. We have Ashwini Singh, who is coordinating this event, Rudrani Datta, Shreya Pujari, Sarthak Tyagi, Nancy Rai, and Surbhi Kumari. They'll be having a live discussion with you, and they have lots of questions for wine preparation and growing grapes. And especially, I'm stuck at fungal diseases of these wines, meaning grape wines, that how do we manage them? But my question will be at the end. So I think I welcome these students, and they want to have a live chat with you about their problems of especially the grape wines. What are the problems and what curiosity is in their mind? So over to the students. You can join.
Good afternoon, ma'am.
Hi.
Hello, ma'am. Ma'am, first of all, I would like to thank you for enlightening us with such an illuminating session. Ma'am, your years of research, your profound understanding of this topic, and your prowess to present it in such a lucid manner had quite an impact on me, and I'm pretty sure that our audience benefited as well.
Thank you.
My name is Shreya Pujari, and I'm a final year student of Sri Aurobindo College. Ma'am, as it was quite evident that fermentation is a key process in winemaking, there's one thing that got stuck in my mind that I would like to ask you personally. So, ma'am, we have... Yes, ma'am. We have seen that the fermentation process in commercial production of wine in large wineries takes months to achieve that desirable taste, aroma, and color. But ma'am, is there a way to optimize this process scientifically to achieve that same taste and texture in a shorter duration of time?
Well, you have seen that a lot of the aromatic substances, compounds are produced when the wine rests. But for a very light wine with a fresh taste and not so rich in aromas, it can be as short as maybe 4 months. But the actual fermentation is maybe it's 2 weeks for degrade most of the sugar that is in the must. And during this time, the wine grower is in the cellar and tests every day, even the taste. And they also use a microscope because there are from the berry skin, there are also yeasts they don't want to have in the wine. So maybe they need to add more of a dried yeast if they have problems with fermentation. And sometimes also fermentation stops. And then they have to change the temperature in the cellar. And well, do you think it would be an advantage to have a faster process?
Ma'am, maybe at commercial level it can be of some advantage, but like what I have realized is that fermentation is the key to that particular rich taste that a wine has, and
Yes,
that aroma
yes.
also.
And
Yes, ma'am.
and you will see it in the price. A very fast produced wine, you can buy a bottle maybe for $2 or $3. And it goes up to $200, $300. It's just the way you want to have your wine. If it's just a drink, fast produced wine would be okay, but for celebration and for really wine tasting or accompany a very special meal, I think we need to have the long process for a good wine.
That's absolutely true, ma'am. Thank you so much, ma'am, for answering my question. Thank you.
You're welcome.
Warm greetings, ma'am. This is Nancy. So firstly,
Hi,
I'd like to thank you for the wonderful insights which you shared with us all about viticulture and enology. So
You're
ma'am, throughout your session, I was wondering that what was your inspiration which basically made you inclined towards enology?
Well, my doctorate professor, he was working on phylloxera because before he started to work on the fish, also you can see genetics go from one organism to the other. And he actually introduced his students to wine and wine tasting. So when I started my education at the university, we did excursions to wine-grown regions, and I got interested. You know, Germany is not so much a wine tasting or a wine, even especially Bavaria, a wine drinking country. We have our wine growing regions where wine is very common, but most of the people prefer to drink beer. And I think I should add back to your question from before, in the German wine growers, they cannot produce these large amounts of wines like California or South Africa and Australia every year with the same taste, but they put their effort in high quality and high price wines.
Thank you, ma'am, for sharing this piece of information. This was really interesting to know. Thanks a lot, ma'am.
You're welcome.
Hello, ma'am. Good afternoon. Rudrani Datta this side. So, ma'am, firstly, thank you for such an informative session. And I'm pretty sure everyone present here must have got a very clear description of both viticulture and enology at the same time. So thank you for that, firstly. So, ma'am, my question is somewhat related on the same production line question, which my colleague Shreya just asked. Ma'am, in your speech also, you have mentioned about the fermenter vessels and the role that they play. So there are various prescribed ways of fermentation. In case of white wine, they have said that oak barrels can be used. In case of red wine, it is either wood top fermenters or stainless steel top fermenters. Ma'am, I was really interested to know if you could distinguish what is the exact function of these type of fermenter vessels on the final wine produced. If you could just let me know that.
There are, of course, in the cellar, you can use all these methods on all types of wines. You can produce red wine like white wine is produced. You will get a wine that is called Blanc de Noir if you press the grapes. It does only work with varieties that don't have anthocyanins in the skin, that only have anthocyanins in the skin and not in the berries. And of course, as you say, people put white wines into oak tanks. It gives them a special taste. You still have the more acid components in the white wines, so you could distinguish. And some people don't like the oak taste in the white wines. And also the tannins, the more bitter tastes you can have in the red wines. And if you want to, this is a commercial thing, if you want to make a wine that will be the same every year, it will be very, very hard to using these different types of methods. But if you want to have a vintage every year, which is different from the year before, where you can taste if the summer was hot or if it was rainy, you can use all the different methods for all the different types of grapes. To make a wine that's the same every year, it's usually not one cultivar, it's cuvées and blending. And they also need chemical analysis for the aromas.
Thank you so much.
Did I answer your question?
Yes, ma'am. Yes, ma'am. It was pretty well clear to me. Thank you so much.
Good afternoon, ma'am. First of all, thanks a lot for such a fascinating presentation. I'm a bit curious regarding the quality of wine present in our local market. Can you elaborate a little bit about more? What are the diagnostic features we should look for while choosing the best quality of wine?
First of all, if you choose the best quality, it will mean you choose the wine you will like best. So it's very different from the personal taste. It's an individual thing, but also there are quality levels. Like table wine would be the wine that is the low quality, and there is Spätlese and Auslese. So late harvest would be a higher quality because of higher sugar content. And this will address also something which has changed with global warming, because of global warming, the sugar content in the berries is higher and higher, which means the alcohol content is also higher. A white wine would be okay with 12% alcohol, but if a white wine has 14 or 15%, it's not drinkable anymore. And late harvest wines will also be sweeter. Picked selection, like the berries are not only the clusters are hand-picked, but also the berries are hand-picked, which are used for fermentation. It would be a selection, a wine selection, or Auslese, you would call it in German. So there will be no damaged berries or no diseased berries in the wine. So this would make it very special. And when you go to you should buy wine where you can taste it. I would offer this, and just don't look. Of course, the price will indicate some kind of the quality, but some wines which are on traded for their price, maybe they don't even taste good anymore because they are aged too long in the bottle.
Thanks a lot, ma'am, for the answer.
Thank you, ma'am, for sharing such valuable knowledge about the topic. As wine industry on such a huge scale, the waste produced must be equivalent. So could you please suggest some ways for waste management?
I didn't understand management for selling or?
No, ma'am, waste management, like the waste from the wine industry. Like ma'am, organic waste, peel of grapes, seeds, water used. Can we use it in organic farming?
Okay. Organic wine growers are, we have a lot of organic wine growers in our region who use these fungal resistant grapes. And they are special, they still have to spray the grapes. And as I already told you, irrigation is not yet allowed. So this will be a problem because the summers are getting drier and we didn't have enough rain during the winters. So if there will be, if the vineyards need additional water, it will be very complicated, especially with our steep vineyards. It's easier to irrigate when it's on a plain. I didn't address the topic, but I had a picture where you can see that in our region, we need to have the inclination of the hills for the sun hours. We grow fruit trees in the bottom of the valley. And what also is a commercial aspect is the areas where you are allowed to grow vines are limited. So there are claims, and the claims are inherited, or you can buy claims for the vineyards. We have lost some wine growing areas because in former times people did side wine growing besides their jobs. Cooperatives have become more common.
Okay, thank you.
Good afternoon, ma'am. This is Ashwini Singh from BSc Life Science third year. Ma'am, I have a question that since these grapes, wines are having some pathogens infections, so then, ma'am, is there any possibility of having some infections in fermentation process also?
Yes, infection in the fermentation process would mean that you have yeast or bacteria that you don't want, and that would make the bad tastes or the bad aromas in the wine. Usually, and also you can have, I've shown you the noble rot, but there are also other rots which would influence the aroma and also influence the fermentation process. And some rots produce vinegar on the grapes. So you don't want to have these berries with the vinegar rot, you don't want to have in your fermentation process. So besides selecting the clusters, there are also optical systems where the clusters go on under a machine, and they check for brown berries and they are discarded. Because you can see you need a lot of clusters for the wine, and it's not, if the machine harvests, you cannot select. The machine just harvests all the clusters. So the this is why the high qualities are also harvested by hand. And there are also other yeast species that you can, some conditions during fermentation prefer these species, and you will have totally different taste after the fermentation. So this is why in the cellar, they also use a microscope and look for like yeast, like Cloqueria, for example. So the students learn to identify under the microscope, and also the Institute offers classes for wine growers where they can learn. And in addition, I didn't mention it, there is also, it still has the name fax, but now it's an email. The Institute gives informations to the wine growers for the vineyards and for the cellars. They have prediction methods for, like this year, we have a special, because of sunburn, there is a special compound, and you have to treat your fermentation berries, maybe add ascorbic acid, which is allowed, and things like that.
Thank you, ma'am. Ma'am, I have one more question. That, ma'am, is it possible to produce wine with 0% alcohol content?
You have to get rid of the alcohol, and there are different methods you can do this, but you lose aroma substances. So it's a different kind of drink, I would say. And
thank you,
of course, you can, a chemist can also do a lab wine, but you wouldn't call it wine. Just put compounds together and make something that would taste like wine, but it's not, it doesn't taste, you would recognize it.
Yeah. So with such a good, thank you everyone. So with such a good question answer sessions, I just have 2 points to make. One of the audiences has asked, are transgenic grapes used in wine making? And if yes, what happens to the quality of wine if it is transgenic?
Okay, we have checked what happens to, well, it has not to be indicated on the bottle, because only if you use transgenic yeasts, which is also a field of making transgenic yeasts with special properties. It has not been indicated on the bottle because you won't find the gene in the product. And we have tested if the fungal, the transgenic grapes that were at we tested at the Institute, they had fungal resistant genes from barley.
Okay.
And we have tested if these genes influence fermentation process, and they don't. And there's also a paper we did on monitoring the fate of DNA during fermentation. And DNA is degraded very, very fast, and you don't find any sequences that were from the transgenes in the must anymore.
Yeah, and then there's another senior colleague of mine from Swami Shraddhanand College, another college in our university. I just tried Google Translate if I could translate. She's saying Guten Tag, sie haben, she knows German probably, she's trying to,
yeah.
She says you spoke very well. I just translated this, and she wants to congratulate you and express her gratitude.
Yeah, I
such
can
a
see
nice talk.
She wrote in the chat, yeah.
Yeah.
So I have spoken very well.
Yes. Yeah. So thank you again for everything here. And I just want everyone to join back for a snapshot. Sarthak is taking a snapshot. And with that, probably we might come to an end of this program. So yeah. So Principal Sir, can you please put your camera on? Sangeeta and all the department colleagues, please join us.
Ma'am, I don't have privilege to turn on my camera.
Yeah, okay, I'll do it. Just hang on 1 minute. So maybe sir has left or is busy with some meeting or what. I requested him, but he's not here. So everybody is in. Where is Rashmi? Rashmi is here, yes. Who is missing? Rahul is not yet joined. Just hang on. Rahul is here. Rahul, put your camera on, please.
I'm here, but I don't have the privilege.
Why you don't have the privilege?
Stop
just give me 1 minute, please. Huh? But you are not there. You have moved out or what? I don't see you. You are the co-host. Please do something for yourself and join. Yeah. And principal sir, probably, I think either he has left for some meeting.
The software is not letting me in my camera.
Manju, I had put one question in the chat box.
I asked just now. Did you not hear the answer? I asked you a question. You missed it. So
about the pollution, I've seen it.
Yes.
Yes.
Yeah. Okay. Yeah.
No, we didn't. We didn't notice the impact during lockdown. The vineyards are not so close to big cities. They are in the river valleys because we need the inclination of the hills for wine growing, and the only wine or vineyards in the plain are in the Palatinate, and there are no big cities. So there was no.
You can get him in.
Yeah.
Thank you.
So you want to try again, Rahul? Join again. Otherwise, I tried my best. I couldn't get you in.
It is not happening. So we should move now.
We should move forward now. So sorry for you, but I don't know. Technology glitches, we can't help sometimes. I tried a lot, but I can't get him in. So yeah, Sarthak, you take your snapshot and then.
I would suggest, can you say cheese, please?
Cheese.
Yeah, smile.
Okay.
Yes, ma'am.
Okay.
Yeah. Thank you, Sarthak. And probably now it's for the students to finish how they are planning to finish this program. So I would give it to them, what they want to do now.
Thank you, ma'am. So firstly, I'd like to request the attendees to fill up the feedback form so that we can get to know that how can we make the future events even more better using your valuable feedback. The link will be in the chat box. Thank you. So as all good things come to an end, so is this webinar. On behalf of Sri Aurobindo College, I take this opportunity to propose vote of thanks to those who made this program a success. At the outset, I thank our honorable speaker of the day, Dr. Angellikaa Scartl, for sparing time from her busy schedule. We are really enlightened with your knowledge and presence, ma'am.
Thanks a lot. It was a pleasure.
Thank you, ma'am. We are thankful to our respected principal, sir, Dr. Vipin Kumar Agarwal, for his enthusiastic support at every step. I'd like to thank Professor Sangeeta Kaul, convener IQAC, and Professor Manju M. Gupta for organizing such a student-oriented, insightful event and their support throughout. I'd like to convey my thanks to the whole Department of Botany, Sri Aurobindo College, for giving us this opportunity to have such an insightful session. A special thanks to the members of the organizing committee, Dr. Rashmi Mathur, Dr. Rahul Dev, and Dr. Isha Gunwal, for their unflinching support and coordination. And heartfelt thanks to the students for their active participation. With these warm words and a kind message, we move to the end of today's webinar. Thank you for being here.
So I just put it off for public, but I like to speak to you, Angela. I want to say thanks to you. Just hang on 1 minute. I'll put it off.
Okay.
Yeah, but we can't do it. Yeah. Again, technological glitches and I can't do it. So my heartfelt thanks once again for sparing your time and efforts. It is just because it was a very good opportunity for our students. They get to talk to us, such a international reputed scientist. Our principal sir is back. So we were looking for you for the snapshot. Sir is there. So Sarthak, can we try it again?
Sure, ma'am.
Yeah. Please, everybody come back. Yeah. Dr. Sangeeta, Dr. Rashmi can also come back. Yeah. So maybe sir would also like to extend his thanks. And the students really felt excited and they really got to know about this science, which probably they were missing at some stage. Sir, you want to say thank you?
Angela, thank you very much on behalf of the college, and on my personal behalf, I would like to thank you for sparing your time and giving a valuable knowledge to our students and to ourselves. I'm not a student of science, but despite of that, I came to know many new things. Which I didn't know earlier. Thank you. Right.
Thank you, sir. Thank you, everyone.
Lecture. Thank you.
Yeah. Thank you, everyone.
Thank you very much.
So with
Thank
this,
you.
we come to an end. Thank you for being here, everyone. Thank you. Thank you. Thank you, Angelika.
Thank you for the wonderful session, ma'am.
Sorry?
Thank you for the wonderful session you have organized.
Thank you, Dr. Angelika. That was really nice. Yes. Thank you.
You're very welcome.
Yeah, we'll meet again, certainly. Thank you.